This is going to be your relatively short video lecture on Chapter 6, which covers viruses. To start us out in this chapter, just some terminology things. So one bacterial cell is a bacterium, one virus is a virion, or virion, I don't care which way you want to say that. Viruses are sort of tricky. They aren't made of cells. Your book calls them acellular pathogens. They show no metabolism when they are outside of a host cell, so they're inert. They can't reproduce outside of a host cell, so there's always sort of been a debate about whether or not a virus is living or not living. When a virus does attach to a host cell through a receptor mechanism, what it can do is insert its genome into the host cell and then trick that cell into doing what the virus wants it to do instead of what the host wanted to do to begin with. So as you can see in this picture, we have a phage, which is a virus that infects bacteria. It docks with the bacterial cell, and it's able to do that because these tail fibers can attach to receptors on the bacterial cell plasma membrane. Once it does that, it injects its genome, which in this case it says it's DNA, but the genome can be RNA in viruses. It's either DNA or RNA, never both. But it injects the DNA into the host cell, and then the host cell starts to read that DNA and do what the virus wanted it to do, which is, in this case, make more little virus babies and then assemble them, and then it's eventually going to end up popping or lysing the cell to release those viruses out into the world. As the host cell is doing this, the host cell is no longer doing what it wanted to do, and so it's sort of been hijacked, and that's how viruses can make you sick, or in some cases even flat out kill you. It's because you're no longer doing what your DNA told you to do. You're doing what the viral DNA tells you to do. All right. Viruses are specific usually to just a couple of a species, although there are some cases where viruses have a very broad host range. But I do want to make sure you understand, like a virus that infects, and I give you the specific example of parvo. Parvovirus is a virus that can infect your dog, but you could full on let your dog lick your face when they have parvo if they could get up to it. And then you would not get parvo from your dog because you don't have receptors that virus can attach to, and a dog does. So that virus can attack a dog, but it can't attack you. Same sort of thing with chickenpox is the example that I'm giving you here. If you were to get chickenpox, your cat could come and lick your face and try to make you feel better, and your cat can't get chickenpox because they don't have the receptors for that like you do. So most species have a fairly narrow range. Some can get very broad. Coronavirus, for example, the one that specifically causes COVID-19, which is SARS-CoV-2. Did I say CARS? SARS-CoV-2. That virus can infect a whole different broad range of species, from bats to pangolins to mink. And that is a problem because they had to wipe out the mink in several Norwegian countries because people could pass it to mink and then mink could pass it back to people. Most viruses don't work that way, though. Okay, your basic anatomy for a virus. We're going to have our genome, which again can be either DNA or RNA. We're going to have a protein coat outside of that that's called a capsid. Now, those two things have to be present in order for us to call a pathogen a virus. So genome, capsid. Then we can have additional things outside of that, or in addition to that, like in the case of a bacteriophage, some of them look like little alien docking ships, and they have a sheath with tail fibers, and the tail fibers are what have the receptors that are going to allow them to attach to cells. Sometimes we're going to have an envelope outside of the capsid, but you got to have these two things in order to have a virus. In terms of naked versus envelope, there's a couple of different implications for this. So this adenovirus that is mentioned up here, this is what's known as a naked virus. It has the genome, it has the capsid, it has spikes on the capsid, but it doesn't have anything else outside of that capsid. HIV, human immunodeficiency virus, though, it has the genome, it has the capsid, and then outside of that, it has a lipid envelope. So naked viruses, no envelope. Enveloped viruses do have that. Now, counterintuitively, enveloped viruses are actually easier to kill because lipids are easier to denature than proteins are. And so we can use disinfectants and antiseptics and things to kill an enveloped virus more readily than we can kill a naked virus. That'll come back to us when we start talking about disinfectants and antiseptics and things later in the book. Okay, viruses, we're specifically going to be talking about phages, which is, again, those are the ones that can affect bacterial cells. they tend to have two different reproductive cycles that they can undergo. They've got a lytic cycle and a lysogenic cycle. The lytic cycle, that lytic stands for lysis. Lysis is when we pop a cell and murder the cell in the process. When a phage is undergoing the lytic cycle, what it's going to do is insert its genome into the host cell. The host cell is going to make a little virus babies, so many of them that the host cell is literally going to burst and release those virions out into the world. So that's the lytic cycle. This cell is now dead. It has burst to release those. So lysis, lytic, those two things go together. A lysogenic cycle works different. This is a phage that's sort of playing a long game because it knows if I kill the host cell, I might not get to spread to anybody else. So I want to bide my time a little bit and make sure I can infect a lot of different things and not just one thing. So we still are going to inject our DNA into the host cell, but instead of making copies immediately, that viral DNA gets incorporated into the DNA of the host cell and it becomes something known as a prophage. Now, every time this cell divides, when it replicates its DNA, it's also going to replicate the prophage. So all of its little baby cells are also going to have that viral DNA inside of it. And so we're getting into all future generations when we put the viral DNA into the host cell. Now, here's the thing about a lysogenic cycle, though. Viruses don't want to stay in a lysogenic cycle forever. What they want to do is sort of bide their time until they know conditions are right. And then when conditions get right, or they can sense that this host cell is about to die, it's going to excise the prophage from the host chromosome and then switch over to the lytic cycle where now the host cell is going to make little viral babies and pop the cell. This act of going from the lysogenic cycle to the lytic cycle is called induction. So lysogenic cycle lasts as long as the host is healthy, but once the host starts to get stressed, we're going to switch to lytic and just go ahead and murder it so that we can make lots of little viral babies. Hold on. Calm down, Captain. Hey, be calm. Shouldn't try to record things while my dog is in a happy mood. Okay. I'm going to pause for a second so he can calm down. All right, I think he's going to be calm now, so we're going to restart this. Animal cells being infected by viruses work a little bit different from how phages can do it. First off, when a bacterial cell gets infected by a phage, the whole thing doesn't enter the cell, only the genome enters the cell. But when it comes to an animal cell, usually the whole virus is going to go ahead and enter the cell, the capsid and all. And so you can see that happening up here. This is a cell that's being infected by the flu virus, which is influenza A, B, whatever letter we want to put after that. So you can see the genome and then the capsid outside of that. And the whole thing comes in the process of endocytosis, usually receptor-mediated endocytosis. After that, we have to uncoat because we can't access the genome when it still has this protein coat around it, so we have to take that off. And so uncoating releases the genome, and then now we can either do lytic or lysogenic sort of thing. We can incorporate that DNA into a host cell chromosome, or we can go ahead and just start the assembly process. From there, things are fairly similar, except at the end, very often, instead of lysing the cell, we're just going to release viruses through exocytosis or secretion. And so that's what's happening off down here. All right. Viral types. I usually use the Baltimore classification system because that's the one I honestly hear get used the most often, but your book does describe a different system. So there's seven different categories that get used in the Baltimore classification system, and it has to do with whether they're enveloped or naked or these 5 things that we got going on up here listed. So what you need to understand here is what the abbreviations stand for, and it is very important that you understand and remember how protein synthesis is going to happen. So for protein synthesis, we've got our genes encoded in double-stranded DNA. Whenever we want to use one of those genes, we transcribe a gene into messenger RNA, and then that messenger RNA finds a ribosome and it gets translated into an actual protein or polypeptide. And so dsDNA is double-stranded DNA. That works just like regular old DNA, so it can do the normal flow, transcription, translation. We get a protein at the end of that. Single-stranded DNA is different, though, because it's just one strand and not 2, and so we can convert that single strand into double-stranded DNA with the enzyme DNA polymerase, and then from there we can use it just like always. Then from this plus and minus, what you got to remember about plus and minus is it's talking about positive sense strands or negative sense strands. A positive sense strand is essentially mRNA, which that's what we make during transcription. So we can translate that directly as is, but a negative sense strand, that's the opposite strand, and we cannot directly translate that. So we have to convert a negative sense strand into a positive sense strand, and then we can go ahead and translate it once we've done that. Now, double-stranded RNA, since it's double-stranded, it has both a positive sense strand and a negative sense strand. The positive strand can be read as is. The negative strand will get copied into the positive strand, and then we can read that as is. Your book spends a little bit of time talking about this RNA polymerase thing that's going on right here. That helps us out with that process. Okay, retroviruses are a special type of virus. HIV happens to be a retrovirus. Retroviruses have this enzyme called reverse transcriptase. So reverse means we're going backwards. Transcriptase is the enzyme that is going to help with transcription, but we're reversing transcription. So where transcription normally goes from DNA to RNA, reverse transcription is going to go backwards from RNA to DNA. So what it can do is convert positive sense strand RNA into complementary DNA that can then help us make lots of copies of other things. And so retroviruses are special because they have that enzyme. One of the other special things that relates to a retrovirus is that enzyme, reverse transcriptase, makes a lot more mistakes than the regular polymerase enzymes that help with DNA replication or transcription. And so that means retroviruses tend to be able to mutate faster than other types of viruses. And that's one reason why vaccines for HIV have been so difficult. It's because that virus evolves so fast that it's hard for us to have a vaccine that's going to prevent it because it just changes before the vaccine can actually make it to market. Okay. In terms of infections, there's two major types of infections, persistent infections and acute infections. Persistent means you're going to have that infection for a long period of time. Acute means you have it for a short period of time. And your book doesn't even talk about acute infections at this point. For persistent infections, there's two subtypes, latent and chronic. For a latent infection, what happens is you get an infection, you get sick, your immune system makes you better, but you did not actually clear the causative agent. In this case, we're going to be talking about viruses. So the classic example for this is varicella zoster virus, which is the virus that causes both chickenpox and shingles. When you get infected by this virus initially, pre-vaccine anyway, you get chickenpox, which gives you flu-like symptoms and makes you insanely itchy. Then you get better though, and you think, yay, it was just an acute infection, but it wasn't. That varicella zoster is now hiding within sensory neurons, and it can come back out later whenever you start to get stressed or your immune system gets weaker. Unfortunately, when it comes back as shingles. Shingles, the best way I've heard it described from people is imagine a part of your body just feels like it's on fire 24/7 and there's nothing you can do to put out that fire. That's what shingles essentially is. So chickenpox is a latent infection because it never went away. It just hid in your body and didn't cause you problems for a while. Chronic infections, once you get symptoms, those symptoms essentially don't go away. You just end up with that disease for the rest of your life. HIV is the best example of a chronic infection, although any of the hepatitis B or C infections are also chronic infections. Okay. In terms of cultivating viruses in a lab, what makes them so difficult to work with is that you can never work with them in a pure culture because you can't grow them outside of a host cell. So you always have to have some other tissue present, and that tissue sort of contaminates the sample because it makes it not pure. An in vivo sample means that you're growing it in living things. So you can grow phages in bacteria, you can grow plant viruses in plant tissue, you can grow animal viruses in like eggs or whatever. In vitro means that you're growing them in like lab glassware. So if you know about in vitro fertilization, that means we make a baby outside of a human being and then we'll try to implant that baby later on. That's basically the same thing here. So one of the labs that we are going to have you guys do involves us cultivating a phage and a lawn of bacteria. So these are viral plates. They are made using what's known as the double layer technique. And so we put down a layer of regular old agar at the bottom of this, And then we mix a soft agar with phage plus bacteria, and then we pour that soft agar on top of the regular agar, and then we smooth it out so that we get this nice thin layer of soft agar with bacteria and phage on it. What will happen is the phage will infect bacteria any place there had been a virus, and so it will kill the bacteria in the spot. And then as the virus reproduces, we kill more and more bacteria so that we end up with this circle of clearing that's called a plaque. So when you see a plaque on a bacterial plate, it means that a virus is on that plate killing bacteria in that one little spot. So each one of these little circles is a plaque, and it shows you a place where a virus got into a bacterium on that plate and then started the process of the lytic cycle on that plate. Okay. When we first started to do this lab, we had to learn some things, which is soft agar solidifies pretty quickly. And so you don't have a lot of time to spread it out on the layer of regular agar on the bottom. And so this, you can see the regular agar that has been spread out right here, all along the bottom. And then as we tried to get the soft agar to spread everywhere, it solidified so fast that we didn't actually get it on here. So you can see the regular agar here. This has the regular agar plus the soft agar on top, and we've got a bacterial lawn that's on top. It's not a very clean bacterial lawn because, again, that agar solidified so fast. We have these solid little chunks of it that are going on right here. Those aren't plaques. That's just we took too long to actually try to spread that soft agar. Okay, from there, your book talks about other forms of pathogens that are acellular. It gets into viroids just a little bit, and then prions. Now, prions are really fascinating and really horrifying all at the same time. Prions are infectious proteins that can basically kill you, is the way prions are going to work. They can infect animals, and if you eat those animals, it can infect you. There's also some genetic forms of it where it just runs in your family and sucks for you, essentially. So PRPC is a normal protein that you have in your brain. Last time I checked, we didn't even know what PRPC was for. What is it supposed to do in your brain? Although I haven't looked recently, but yeah, we don't even know what its main function is. Now, when they were first researching prions, the disease they were working with was scrapie. Now, scrapie is an encephalopathy that's present in sheep, so it's not even one that usually messes around with people, although it can. So when they discovered that this protein was causing the disease, it was called the scrapie protein. So that's why it's called PRPSC. So this is the prion protein. Now, I'm going to try to relate this to a zombie because we all know a zombie used to be a person, and then something happened, and that person became a bad brain-eating person. Well, that's how PRPSC works. It had been a normal, happy, everyday protein, and then it got misfolded and it became a zombie. And what it does is it goes and it looks for normal protein to bite, and then it bites it and it converts it to zombie protein. So PRPSC is just running around zombifying what had been a normal protein and turning it into a bad protein. And as it does that, the accumulation of bad protein ends up creating these holes in your brain. It's literally eating your brain. And of course, if your brain is gone, you are gone. And so it ends up invariably killing you if you have this. We have no treatment for prion diseases. Again, it basically just sucks if you get it. We can watch you die, and that's about all we can do for you if you have it. There are a few different diseases in humans that are prion diseases. Your book spends a little bit of time talking about, and I always say this in the American way, Creutzfeldt-Jakob, but it's actually, I think, named after a French doctor, so it should be like Creutzfeldt-Jakob or something like that. But that disease, it's sort of like Parkinson's and Alzheimer's and depression, schizophrenia, and anxiety all rolled in together until you die. And there's different ways that you can get CJD. There are some people who just had the bad luck that their DNA polymerase mutated the gene for PRPC and now they have PRPSC. There are some people who it runs in their family and they know there's a 50% chance that it's going to happen to them. There are some people in Britain who got it because they ate beef during the mad cow epidemic that happened back in the 1990s. Here in Texas, we have this issue with something called chronic wasting disease, which is seen in deer. And hunters have to receive educational classes prior to getting a hunting license in Texas. And one of the main things that they are taught during that is that if they find a deer that is wasting away and it looks really skinny, but the other deer in the area look relatively healthy, that deer might have chronic wasting disease. And if you were to eat that deer, you might develop CJD from that, because chronic wasting disease is a prion disease. And so it's possible, although there haven't been documented cases of that yet. Prion diseases, however, do take a long time to develop. So it's possible that some people might end up developing that disease eventually. Then I also wanted to spend a little bit of time talking about what to me has to be the worst disease in the history of the world, which is fatal familial insomnia. And I feel that way because I frickin' love sleep. Sleep is like a vacation for me at the end of every day, and I look forward to it, and I dread getting out of bed, and I just love sleep. Fatal familial insomnia is you get this disease that runs in your family. You've watched a family member slowly go insane until they died because they lost the ability to go to sleep. Sleep is such an important part of your brain's overall health. It is a time for you to process memories. It is a time for you to alter the flow of cerebrospinal fluid in the brain so that you could clean out brain tissue of unhealthy protein aggregates. It's just really important. And if you lose the ability to sleep, your brain can't do any of those things anymore. So you can't form memories, you can't clean your brain out, and your brain just deteriorates and makes you go insane, and then it kills you. And so I give you guys 2 different chances for big extra credit opportunities in this chapter. One is The Immortal Life of Henrietta Lacks, because this chapter does talk about tissue cultures and cell lines, and it mentions Henrietta Lacks in there. And then the other is a book that describes, again, what to me is the worst disease ever, fatal familial insomnia, which is, what was it called again? The Family Who Couldn't Sleep? I gotta look. It'll take me a second to find it on my bookshelf. Yeah, The Family That Couldn't Sleep by Max. It's an interesting book, and it's a story that most Americans have absolutely no idea that there are families that suffer from this, and so it's just an interesting book to read. But it's a prion disease, and so I wanted you guys to have that as an opportunity to learn more about prions. Hopefully that helps you get through some of the tougher material in this chapter, some of the more interesting material in this chapter.